inactivation of Escherichia coli 0157H7 in apple juice and apple cider by triamcinolone. So, E. coli has become a significant foodborne pathogen in the United States and is generally found in undercooked ground beef and raw milk, as well as other things such as lettuce, spinach, and even water. So in recent years, unpasteurized as well as pasteurized fruit juices have been found in multi-state outbreaks that have been associated with E. coli. However, the low pH of apple juice and apple cider make them a target for E. coli contamination because of such things as drop or windfall apples. Those are the apples that are found maybe on the ground, and animal feces and fertilizer. So consumers these days are becoming more aware of food ingredients, and they have become less likely to purchase these foods that contain synthetic preservatives, and they want more natural preservatives and are less accepting of these commercial preservatives. So the food industries as well as producers are looking for alternatives to the chemical preservatives. So they are looking at things such as essential oils from plants and herbs to find antimicrobial properties. So the objective of this study was to investigate the efficacy of low concentrations of triamcinolone for inactivating E. coli 0157 in apple juice at storage temperatures for room temperature at 23 degrees Celsius and refrigeration temperatures at 4 degrees Celsius. So they got five-strain cocktail of E. coli 0157 and used it in the study. So they got the cocktail from milk, meat, calf, apple cider, and apple juice isolates. And what they did was they cultured the strains, centrifuged them, washed them, and plated them, incubated them for 24 hours, and to obtain a bacterial count, and plated them on TSA plates. And they used the triamcinolone or TC concentrations ranging from 0.025% on to 0.125%. And for sample preparation, they purchased apple cider as well as apple juice from local grocery stores. They tested those for the presence of E. coli already in those juices, and they inoculated those samples and added the cocktail of the triamcinolone and took those samples and incubated them at room temperature as well as refrigeration temperatures, and stored those the apple juice at 4 and 23, which was room and refrigeration, but then the apple cider was stored at refrigeration temperatures. So for microbial analysis, the E. coli 0157 in the apple juice and apple cider was sampled on days 0, 1, 3, 5, 7, 14, and 21 by plating. And they used the streaking method to confirm the colonies by streaking on SMAC or sorbitol McConkey agar. And they used a latex agglutination test also for confirmation. And they used a pH. The pH was measured by determined by using a Acunet pH meter also on those days, the 0, 1, 3, 5, 7, 14, and 21. And the addition of the triamcinolone, it did not show any significant change in the apple juice or apple cider as far as pH was concerned. So for statistical analysis, they the data from the independent replica trials were pulled together and analyzed using a general linear model for statistical analysis. There's a particular software that they use for that. So for the results, there was no inactivation, there was inactivation of E. coli 0157H7 in the apple juice that was stored at room temperature, and the transcendentaldehyde concentrations at 0.125 and 0.075% completely inactivated the E. coli on days 1 and 3 of storage, and the 0.025% reduced the E. coli to undetectable levels on day 5, meaning that there was no E. coli detected on day 5. In the control sample, it decreased gradually and tested negative for E. coli on day 14. So in the populations of the E. coli slightly declined in the control of the juice and the cider over the entire 21-day storage period. So the E. coli populations in the treated juice and cider were reduced to undetectable levels on days 3 and 5, as well as 14, by the concentrations of 0.125 as well as 0.025 of the concentrations of transcendentaldehyde. So the inactivation of the E. coli in apple juice and cider by transcendentaldehyde was found to be dependent on the concentration of the transcendentaldehyde. So it did show that the transcendentaldehyde was effective in reducing the amount of E. coli that was in the juice as well as the cider. But the temperature also played an important role in the inactivation of the pathogen. So the antimicrobial effect of the transcendentaldehyde at room temperature could be attributed to the metabolic growth and death rates of the E. coli at room temperature compared to the refrigerated temperatures. So the results indicate that the low concentrations of transcendentaldehyde may potentially be used as an effective antimicrobial to inactivate E. coli in apple juice as well as apple cider. So as far as future work is concerned, they may want to adjust the amounts of transcendentaldehyde that was added to the apple juice and apple cider to maybe add to the sensory effects of it. So that maybe sometimes, a lot of times, maybe during the holidays, cinnamon is added to apple juice. So to have, or apple cider to give it a more flavorful effect, or even a kind of festive flavor. So they may want to play around with the formulations a little to give it more of a flavorful taste, and then even evaluate lower concentrations of transcendentaldehyde to see if it will inactivate the E. coli in the apple juice and apple cider as well.
Kendra, what's the basic difference between an apple juice and an apple cider?
Basically, from my understanding, it's the way that it's processed. So what I've read up on is the apple juice. I want to say they're both pasteurized. I thought in the beginning when I first started researching it that apple cider wasn't pasteurized, but now I'm seeing that they both are pasteurized. So I think now it's more so that
you drink apple cider during the holidays?
I don't.
Have you seen people drink or have
It,
you?
it seems that it seems thicker to me, but I'm not sure as far as the processing of it stuff. But I do know that it's processed differently. I just don't know exactly how it's processed.
Okay. You mentioned E. coli 015787. What does the 015787 stand for?
That is the serotype of the pathogen. But what it stands for, I don't know.
Okay.
I know that there are different strains of E. coli.
Now, the authors of this paper used trans-cinnamaldehyde. Now, if you were to get another compound from cinnamaldehyde, it would be a cis, possibly, it could be a cis, right? What's the basic difference between a trans and a cis compound?
I know that they're like mirror images of each other, or that one is on one side and one is the other side. So they're like facing each other, or like on the compound. So was it like an isomer, like a?
Which is on which
I
side?
may be saying that wrong.
How does trans differ from cis? What is an essential oil you
Cis
mentioned?
is cis like on the same? Is cis the same? Trans opposite? Is that how it goes?
What is on the opposite and what is on the same?
Is cis the same? And trans on the opposite? Is that?
No, what is on the opposite? When you say cis is on the same, what is same for cis? What is on the same side for cis? Last question, what is an essential oil? You mentioned an essential oil. You mentioned some of the spices like cinnamaldehyde may have essential oils that are used for as a preservative. What is an essential oil?
The oil that comes from the plant, like the oil that they derive from a plant, like oil from a clove, or an oil from cinnamon, or oil from
What does it contain?
the basic elements from that particular plant, like
All the elements? Okay. Good. Thank you.
You went through and you said that there was a general linear model. What is a general linear model? What is that?
the statistical software.
Okay,
That what you're talking about?
sure. What is that? What does that do?
I'm not familiar with the software that they use.
How does trans-cinnamaldehyde work?
It's an antimicrobial agent. As far as I know, it's an aldehyde, and that's really all I know about trans-cinnamaldehyde. That it's an aldehyde, and it's taken from the bark of the cinnamon plant.
Okay, so how does an aldehyde serve as an antimicrobial?
I would assume that an aldehyde is like an aldehyde is similar to like a want to say like an alcohol or something, but I'm thinking along the wrong lines. But because it's antimicrobial, it's breaking like the cell wall or the membrane of the E. coli and weakening it.
You went through and you said that future work would need to go through and look at sensory, right?
Mm-hmm.
How would you recommend, or how would you set up an experimental design to go through and look at that sensory work?
I guess I would test different, the way that they put in different percentages of the transaminaldehyde. I guess I would lower those levels or percentages of it and test those to see how those percentages, I guess, fare, to see how people, I guess, rated them, to see as far as flavor, how they like the flavor of it, to see.
Why would you not use the percentages that they use during their experiments?
Well, because obviously those flavors, those seem to fare well, but to see if other percentages would work as well, lower percentages. Because if a lower percentage worked, then you may not need to use as much.
So you're saying that they did not use the minimum amount that would inactivate E. coli?
It's possible.
So if you went through and you set up another experiment, knowing what you know from experimental design and things like that, courses like that, how would you go through and set up that experiment? What would be your treatments?
Let's see, since they I guess they started at around 0.25 or 0.025. Then I may start at around 0.010 maybe, and go up from there. And maybe my maximum, instead of my maximum being 0.125, then my maximum maybe my max would be 0.75, and set it up like that with my minimum starting around 0.10 and going up to 0.75, and pretty much set it up the same way, but lower my minimum and lower my maximum and go from there. Because I'm basically doing some similar research in the lab myself. I have my experiment set up the same way, but I'm not adding anything to my research. I'm inoculating my apple juice with E. coli, but I don't have a cocktail. I just have one strain of E. coli. And I inoculated the apple juice and I set it up to test on similar days.
Going through and looking at E. coli 157H7, how does that get transferred to the drop apples?
I guess after they drop from the tree and drop onto the ground could be contaminated. The soil could be contaminated. So from my research, they're not allowed to use apples that have been dropped to the ground. They're not allowed to use dropped apples anymore.
So how is this still a problem? If they're not allowed to use drop apples?
Then because they're not allowed to use it, then it's becoming less of a problem in pasteurized apple juices. So the articles that I've been finding lately have been more so with problems in unpasteurized apple juice. It's not so much a problem in pasteurized juices anymore.
what do you believe to be the source of contamination?
More so, as far as juice is concerned, I think it's more so with, maybe a problem with either soil from other contaminants, or possibly cross contamination, or even, maybe mishandling.
Any other questions?
Do you think, or is there, as far as cross-contamination, do you think that there is a hazard plan in place wherever it's taking place, or there's not enough, CCTVs in place to ensure that the processing of the apple juice or apple cider is
I think that all companies have to have a hazard plan in place, from my understanding. Every plant or company has to have a hazard plan in place. But the hazard plan doesn't necessarily ensure that contamination doesn't happen. It just has it in place so that if something does happen, they have a plan in place to have corrective measures to take care of it when something does happen. It's like this, this recall that just happened with Bluebell ice cream. They must have had something in place, but they had the recall, so they had to recall everything once it did happen. So they have something in place to take care of it once it happens. But the hazard plan is about safety, but in order to ensure that safety, you have a plan of action in place once, or if something does occur. So, but it doesn't necessarily prevent something from occurring.
All right, thank you very much.